All right, we are in lesson 2 of our genetics unit. Today, our focus is on DNA replication, which is the process of making more DNA. So remember, DNA contains the instructions for making proteins, and the cell's like a little factory that's just cranking out those proteins. But in order for these instructions to be used, they first have to be replicated. So that's what we're talking about today. All right, so let's talk about the where and the when, and then we're going to get into the how. So let's start with the where. Where does this process of DNA replication happen? So in eukaryotic cells, remember that cells with a nucleus, DNA replication actually happens in the nucleus. I do also want you to recall that not all cells have a nucleus. So prokaryotic cells, for example, do not have a nucleus. And prokaryotic cells undergo DNA replication as well, but it's a little different than with eukaryotic cells. Our focus today is on DNA replication in eukaryotic cells. So these are cells with a nucleus, and DNA replication happens in the nucleus. Okay, so that's the where. Now let's talk about the when. When does this process happen? Now this is not new to you because in our cells unit, unit 2, we talked about mitosis. And mitosis is the process of taking a parent cell and turning it into 2 daughter cells that are genetically identical to the original parent cell. Now, in order for this to happen in each daughter cell to get the same DNA that the parent cell had, we have to replicate that DNA. And so that process happens right before mitosis in what we call S phase. S phase of interphase. So we have interphase, and then we have mitosis, and then we have cytokinesis. And so this process is going to happen in S phase of interphase. All right, now we can talk about the how. So we're going to start talking about the process of how DNA replication happens. I do want to remind you from the last lesson that DNA has what we call nitrogenous bases. There are 4. So we have adenine, thymine, cytosine, and guanine. And there is something that we discussed in the last lesson called base pairing rules. And we said that A, or adenine, always pairs with T, which is thymine. And C always pairs with G, so cytosine always pairs with guanine. And you'll need to remember these base pairing rules for today. So I gave you an example in the last lesson where I gave you half of a strand of nitrogenous bases for DNA, and then I asked you to use base pairing rules to find the complementary strand. And so here we have adenine, guanine, thymine, cytosine, adenine. So if we use base pairing rules, we might get something like this for our complementary strand. And again, A always pairs with T, C always pairs with G. All right, now another thing that I want you to get in your notes is this descriptive term that we give for DNA and its ability to replicate. So we say that DNA is semi-conservative because replication is going to use existing DNA as a template to make a new strand of DNA. And so this will make more sense as we get into the lesson today, but I just want you to hold on to the fact that semi-conservative is the adjective that we use to describe a DNA molecule. All right, so this image shows the process of DNA replication, and you can see here that you have what we call an original strand of DNA. You can see the nucleotides pairing there using base pairing rules. What's going to happen in DNA replication is this original strand is going to be split. And so you can see the original strand on the left and the original strand on the right. And then we're going to have some enzymes that are going to come in and build a new strand using base pairing rules. And so because we have the original strand paired with the new strand, this is where we get the term semi-conservative. All right, another thing that we mentioned in the last lesson is that these nitrogenous bases are held together using hydrogen bonds, which are weak bonds. This makes it really easy for DNA to replicate. All right, so copying DNA requires many different enzymes, which are proteins. We talked about that in Unit 1, but we're going to focus on 3 key players today. So helicase, DNA polymerase, and then ligase. So helicase is going to serve as a an enzyme that separates the DNA strands. So let me go back to that picture. Okay, so you can see the original DNA strand is going to separate, and the enzyme that's going to actually unzip the DNA molecule is helicase. And then DNA polymerase is going to be responsible for adding nucleotides. It's like the builder. So as DNA replicates, it will build a new strand of DNA by adding nucleotides. Now, DNA polymerase is also going to proofread, which is really important. We're going to talk about in our mutation lesson later in the unit the process of proofreading and what happens if DNA polymerase misses something. And then we have the enzyme ligase, which is going to link together those new pieces of DNA. All right, this is a good visual representation of sort of what happens and how the process of DNA replication works. So DNA replication is going to start at a certain point. We call this the origin. And the origin is usually identified by certain DNA sequences. But at the origin, helicase is going to come in. You can see that here, and it's going to unwind or unzip the DNA. And then DNA polymerase is going to come in and build a new strand of DNA, specifically by adding bases to the new strand. And then ligase is going to come in and just sort of seal the new pieces of DNA together. And so when we're finished, DNA replication yields two identical double helix DNA molecules. And it's going to be from that one original double helix DNA molecule that we started with. But just some fun facts about DNA replication. So it takes cells around 6 to 8 hours to replicate DNA. And by the time you are watching this video in your lifetime, your cells have replicated trillions of times. Now, we did mention DNA polymerase. It does proofread during this process, and it does an amazing job of proofreading during DNA replication, but it does sometimes make mistakes. So if it does happen to match the wrong DNA bases, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein or no protein at all. But we're going to discuss that whole process in our mutations lesson. So we'll save that for a future lesson. All right, so that ends our lesson over DNA replication. And I will see you in the next lesson where we're going to talk about protein synthesis.